So welcome again in another video lecture from protein engineering. So today we are going to discuss about an interesting discovery by protein engineering that saves lives of many smokers. So that discovery is the development of the oxidation resistant variants of alpha-1 antitrypsin. Okay. So first of all, I am just providing this notes like things that I have written, and I will going to explain this via some diagrams and flowcharts. So if you want to make a notes on the basis of this slides, so I would like to, so now I think it will be easy for you to take screenshot. Okay, so now let's start our discussion. So first thing I want to discuss with you that during respiration, are we only breathe pure air? No, obviously not. We, along with air, we are breathing various kind of dust particles, along with, most importantly, various kind of microbes, right? That is why for various microbes the respiratory tract is an important portal of entry, right? And via breathing, the microbes who get entry into the body will obviously be transported to the lungs. Okay, and microbes, as its basic nature is causing some harm in the body, so, and as it is migrating towards a vital part of our body, so our immune system should have some defense mechanism against it. Obviously, yes, it is obvious. So there are various kind of immune mechanism, but that is not our point of discussion today. We will going to discuss about only a one kind of immune mechanism that is operating in the lungs. Okay, although there are various kind of immune system present during the journey of the microbes from the environment to the lungs. Okay, so for that reason body knows the situation, that body knows that there will be too much amount of microbial population migrating in my lungs. So that is the reason why they have activated constant circulation of leukocyte and lymphocyte. This leukocyte and lymphocyte just like patrolling police. Okay, they are constantly patrolling towards the lungs. Okay, so among those leukocytes there is an important leukocyte known as neutrophil. Okay, I know you know about the neutrophil. So they also migrate towards the lungs, and inside the lung they produces an enzyme called elastase, and this elastase has two kind of activity, let me tell you. So the first activity is it will control the microbes. Okay, and second one is it will degrade the elastase protein of the lungs. So let me tell you that the first activity that it controls the microbes is the positive activity of the elastase in terms of body. That means it helping our body to survive, right? But the second one is a negative activity in terms of our body. Why? Because remember, the elastin protein in the lungs is responsible for controlling the elasticity of the lungs, and as it is destroyed, the elasticity of the lung will fall. Okay, and that is not a good news for the lungs. Okay, so as it is a natural phenomenon, our body should develop some defense mechanism against it, and yes, body have a defense mechanism against it. So what kind of defense mechanism it has? So for countering the negative effect of elastase, body have a special type of protein in its serum known as alpha-1 antitrypsin. Okay, This alpha-1 antitrypsin have the ability to counter the negative activity of elastase. How we will going to discuss, but let's discuss the topic first broadly, then we will going to enter in the details of it. Okay, so that means whenever microbes is inside the lungs, our neutrophil produces elastase. Okay, clear. And this elastase have two kind of activity. One activity is it will control the microbial population. Okay, that is obviously no doubt a positive activity in terms of our body. And the second activity is it destroys the elastin protein. This elastin protein is responsible for the elasticity of the lungs, and as it is destroyed by the elastase, the elasticity of the lung will fall. Okay, so the negative effect is countered by another serum protein that is known as alpha-1 antitrypsin. How we will going to discuss, don't worry about this. So that is a almost win-to-win situation for our body, right? But there is a twist in the whole story. So I hope you all know that neutrophil also possess another type of killing mechanism known as oxygen dependent killing mechanism. Okay, so here the spelling of oxygen is quite wrong, that the spelling will be oxygen dependent killing. So sorry for that. So this oxygen dependent killing produces reactive oxygen species commonly known as ROS. Okay, these reactive oxygen species not only control the microbial population, but it also exhibit a negative activity. So what kind of negative activity it shows? It reacts with the alpha-1 antitrypsin and reduces the counter activity that alpha-1 antitrypsin shows to prevent the negative activity of elastase. Okay, that means this ROS helps elastase to show its negative activity by reducing the countering power of alpha-1 antitrypsin. Okay, so that is the big picture of the entire concept. That is, in our lung, whenever there is a microbes, the neutrophil will produce elastase. This elastase have the ability to control the microbes. Not only that, it also have the ability to destroy the elastin protein. Okay, and as it destroying the elastin protein, the elasticity of the lung will fall. Okay, now under such circumstances, our body has developed its own defense mechanism. So what kind of defense mechanism it shows? So remember, mind it very carefully, that there is another serum protein called alpha-1 antitrypsin. This alpha-1 antitrypsin counter the effect, that is the negative effect of the lung. Okay, now under such circumstances, neutrophil also shows its oxygen dependent killing pathway. That killing pathway not only controls microbes, but also the ROS produced during the oxygen dependent killing pathway controls or down regulates the countering activity of the alpha-1 antitrypsin. Okay, and as a result, elastase now can cause the degradation of the elastin. Okay, so that is how ROS is helping the elastase to show its negative activity. Okay, so now I want to discuss with you how this alpha-1 antitrypsin counters the negative activity of elastase. Okay, so once elastase protein performs its positive activity, because remember the negative activity is the after effect. So once elastin protein performs its positive activity, that is the controlling the microbial population, our alpha-1 antitrypsin comes in front of elastase. So I want to told you that alpha-1 antitrypsin is a 394 amino acid protein. Okay, and among these 394 amino acid, there is a methionine in the 358 position. Sorry for that. And there is a serine in the 359 position. Okay, so whenever elastase faces this alpha-1 antitrypsin, this elastase recognizes the peptide bond between 358 methionine and 359 position serine. Okay, and facilitates a formation of association. Okay, that means it facilitates a association. After that association, it will cleave the peptide bond between the 358 methionine amino acid and 359 serine amino acid. Okay, I am again repeating that after elastase performed the positive effect, that is the controlling the microbes, as a after effect it starts to show the degradation of the elastin. Okay, under such circumstance, the serum protein alpha-1 antitrypsin came in front of elastase. Okay, and elastase recognizes the 358 methionine residue of the 394 elastase amino acid containing elastase and the 359 position of the serine. Okay, and they form a association with the elastase. That means there will be a complex of elastase and our alpha-1 antitrypsin. Okay, after that association, the elastase cleaves the peptide bond between 359th amino acid and 358 methionine. As you can see that there is no peptide bond, right? So it is cleaved by elastase. Now after that cleavage, researchers found that once the cleavage is completed, the chance of dissociation of elastase from the complex form of alpha-1 antitrypsin and elastase is negligible. Okay, and as a result, alpha-1 antitrypsin here act as a inhibitor. Okay, by forming a complex with elastase. Okay, so that is the overall mechanism. So under such circumstances, Ross reacts with the 358 position methionine. Okay, and convert methionine into methionine sulfoxide. Okay, so the certain conversion of the methionine into methionine sulfoxide causes a problem. What kind of problem? Let me discuss. That whenever the 358 position methionine is converted into methionine sulfoxide, which is a bulkier group compared to methionine, the alpha-1 antitrypsin can no longer inhibit elastase so strongly as it can in the normal situation. Okay, that means Ross converts methionine into methionine sulfoxide, which is present at the 358 position of the 394 amino acid elastase. Okay, and convert it into methionine sulfoxide, which is a bulkier group. Okay, so under such circumstance, when there is a methionine sulfoxide at the 358 position of the alpha-1 antitrypsin and serine in the 359 position of the alpha-1 antitrypsin, now this alpha-1 antitrypsin, that is modified alpha-1 antitrypsin, I should say, I can say this, cannot be able to inhibit this elastase so strongly as it can in its normal form, and as a result, now elastase cannot be inhibited by the alpha-1 antitrypsin, and as a result, elastase now have the ability to degrade elastin, which falls the elasticity of the lungs. So that is the concept how the entire thing is going on. Now I want to told you that in case of smokers, the situation that I have mentioned here is predominant, because the more they smoke, the more there will be the increased amount of leukocyte population in the lungs. Okay, and along with leukocyte, it is obvious there will be increased amount of neutrophil. Okay, so neutrophil will continue to produce elastase. Okay, that means there will be a relatively higher concentration of elastase. Not only that, the concentration of ROS will also be very high. Okay, as compared to them, the amount of alpha-1 antitrypsin is relatively low, because they are not affected by smoking, right? Under such circumstances, ROS have the capability to convert all the alpha-1 antitrypsin present in the serum into that modified alpha-1 antitrypsin. That means that methionine sulfoxide containing alpha-1 antitrypsin. Now, this modified alpha-1 antitrypsin, which is a very poor inhibitor of elastase, cannot counter the negative activity of the elastase. And as a result, the elasticity of the lung for those smokers starts to fall significantly, and which facilitates a complication known as emphysema. Okay, this is, I hope you know about emphysema. The spelling of emphysema is emphysema. So this is the major medical complication that smokers absorb, and that is for that reason. Okay, so I am again repeating what is going on in case of smokers. So the larger they smoke, the larger there will be leukocyte and lymphocyte population in the lungs, because immune system have to protect the lungs, right? That is why they increases the circulation of the leukocyte and lymphocyte, and along with leukocyte there will be increased population of neutrophils. Okay, and as there is a too much amount of neutrophil, so it is obvious that there will be too much amount of elastase. Okay, and along with that there will be also too much amount of ROS. Okay, in compared to them, the concentration of alpha-1 antitrypsin becomes relatively low, because it is not affected by the smoking, right? It is present in actual amount what it is present in its normal form in the serum, right? So now this small amount of alpha-1 antitrypsin can be easily converted into modified alpha-1 antitrypsin, that means methionine sulfoxide containing antitrypsin, with the help of by the ROS. Okay, as a result, this modified alpha-1 antitrypsin, who are the poor inhibitor of the elastase, cannot counter the negative activity. Okay, so under such circumstances, the elasticity of the lung becomes significantly fall, which is a medical complication known as emphysema. Okay, and that is commonly observed in case of smokers. Under such circumstances, our recombinant DNA technology, along with more specifically saying the protein engineering, came into the picture. Okay, so scientists decided to do about something, or to derive some solution of that problem. So let's see how they solve that problem. Okay, now let's try to focus on the structure of the alpha-1 antitrypsin. So in the very first video of application of recombinant DNA technology, I have discussed about the concept of site directed mutagenesis, right? That with the help of that we can change a single amino acid. Okay, so here the scientists are using the concept of site directed mutagenesis to convert methionine into valine. Okay, that is a single codon change. There is a codon for valine known as GUG, and we all know that universal codon for methionine is AUG. So they are changing the A into G, converting the methionine into valine. okay, with the help of site directed mutagenesis. And if you don't know what is site directed mutagenesis, how it is performed, then I will recommend you to go and watch, and I will also put the link of the video of site directed mutagenesis in the description box. Okay, so that will facilitate the conversion of methionine into valine. Okay, now what is the utility of converting methionine into valine? Let me tell you. So it was observed that elastase can still recognize the peptide bond between valine and serine, okay, and as a result it become inhibited. On the other hand, it was also observed that the ROS cannot convert the valine into methionine sulfoxide, okay. ROS simply cannot do anything to valine, okay. As a result, valine become intact. So this recombinant form of alpha-1 antitrypsin, which is known as oxidation resistant variant of alpha-1 antitrypsin, because it has the ability to prevent the oxidation caused by ROS. That is why our elastase becomes permanently inhibited, and as a result now it cannot do anything with the elastin protein of the lungs, okay, and as a result the elasticity of the lung remains intact. Okay, so that is the concept how protein engineering protects us from the emphysema. Okay, so now whenever there is an individual who have complications like emphysema, the doctors will inject this recombinant form of alpha-1 antitrypsin, that is oxidation resistant alpha-1 antitrypsin, in their blood. So there are now oxidation resistant alpha-1 antitrypsin in their serum, which can not only inhibit the elastase enzyme but become intact even the presence of ROS, because ROS cannot do anything to valine. Okay, so that is the concept regarding the oxidation resistant variants of alpha-1 antitrypsin. So I am again repeating the entire story, that yes, we are respiring microorganism along with air, and as a result microorganism getting direct entry into the lungs, although they have to face various kind of immune barrier before that, but we are not going to discuss about this in that video. So under such circumstances our immune system also activate a continuous circulation of leukocyte and lymphocyte, okay, and among those leukocyte there is a increased amount of neutrophil present, okay. So whenever neutrophil enters into the lungs, they convert neutrophil, they produces an enzyme called elastase. This elastase has two activity: one, they can control the microbes, and second is they can degrade the elastin protein of the lungs, okay. So under such circumstances the elasticity of the lungs will significantly fall, okay. Now not only that, our body have normal defense mechanism against this, that is the serum alpha-1 antitrypsin protein that can counter the elastase, okay. But neutrophil also have oxygen dependent killing pathway, okay, and during that killing pathway it produces ROS, that is reactive oxygen species. These reactive oxygen species have the ability to reduce the countering activity of the alpha-1 antitrypsin. How? Let's discuss. That alpha-1 antitrypsin is a 394 amino acid, and in that 394 amino acid there is a methionine at the 358 position and serine at the 359 position, okay. So whenever elastase has completed its positive activity, the alpha-1 antitrypsin came in front of elastase, okay, and elastase recognizes the peptide bond between 358 position of methionine and 359 position of serine, okay, and there is a association between elastase and alpha-1 antitrypsin, okay. After that association, they will cleave the peptide bond between 358 position of methionine and 359 position of serine, and researchers found that after that cleavage, the dissociation of elastase from that elastase alpha-1 antitrypsin complex is negligible, and as a result elastase becomes permanently inactivated. Under such circumstances, ROS came into the picture and it converts the 358 position methionine into methionine sulfoxide, okay, and which converts alpha-1 antitrypsin into a poor inhibitor of elastase, and as a result elastase can no longer be inhibited by the alpha-1 antitrypsin, and now elastase have the ability to affect the lungs, okay. And this situation is predominant in case of smokers, because the more they smoke, the more there will be leukocyte lymphocyte population in the lungs, okay. So under such circumstances there will be obviously the higher population of neutrophil, that means higher concentration of elastase. Not only that, that means higher concentration of the ROS, okay. That means, and in relative to them, the alpha-1 antitrypsin become negligible in amount, and the all the alpha-1 antitrypsin present in the serum will be converted into modified alpha-1 antitrypsin, that means the methionine sulfoxide containing alpha-1 antitrypsin, okay. So under such circumstance, the alpha-1 antitrypsin, that is the modified alpha-1 antitrypsin, cannot no longer inhibit elastase, okay, and as a result the elasticity of the lung significantly fall, which is a medical condition called emphysema, okay. So under such circumstances the protein engineering came into the picture, where researchers with the help of site directed mutagenesis changes the 358 position methionine into valine, okay, and as a result it was observed that this modified oxidation resistant alpha-1 antitrypsin can inhibit elastase, okay, but this modified oxidation resistant alpha-1 antitrypsin is now no longer be converted by ROS, okay, and as a result they can stably inhibit the elastase, and as a result the elasticity of the lungs become unaltered, right. So nowadays whenever emphysema patient in the hospital, doctors try to inject that oxidation resistant alpha-1 antitrypsin that can control the fall of the elasticity of the lungs by inhibiting the elastase of the neutrophil, okay. So that is how the entire thing is going on. So I hope you have understood the concept of oxidation resistant variants of alpha-1 antitrypsin, why it is important, how it is constructed, and why and what is the story behind it. So I hope this video will be helpful to you. So thank you for listening to this video lecture, and if you find it helpful please subscribe to my channel, like this video and share with your friends, okay. So thank you, bye bye.